organizers for giving us this opportunity. So my name is Matt Akamatsu, and Eran and I are brand new assistant professors. We both started our groups this summer. Thank you. We started this collaboration as late postdocs, and so now this is one of our first projects together as in our new groups. So I'm in the biology department at the University of Washington, and Eran and I recently started a collaboration to use multiscale modeling to understand how actin networks produce force during mammalian endocytosis. So first, I'm going to provide an introduction to the biology and version 1 of our biophysical model. Eran will talk about his integrative framework for integrative modeling, and then we'll both get back onto the stage to talk about our initial steps towards putting these two things together. So the part of the project that I'll describe is the result of a collaboration that was interdisciplinary largely across two groups. Padmini Rangamani's group, which is a theory group at UC San Diego, and David Drubin's group, which is a cell biology lab at UC Berkeley. In this network diagram, each line represents a regular interaction that occurred between researchers during the pandemic over Slack and Zoom, whose expertise ranged from theory to modeling to fluorescence microscopy, biochemistry, electron microscopy. And this feedback, partly enabled by the pandemic, led to a really satisfying feedback loop between modeling and experiments, which allowed us to interrogate our favorite cell biological mechanisms. So we studied the biophysical mechanism by which the actin cytoskeleton produces force to shape cellular membranes. Our long-term goal is to help identify some of the principles that govern the transition from these non-living protein complexes and membranes to living and adapting cells. In this lattice light sheet microscopy movie of a migrating cells, we can see the range of shape changes that occur on a plasma membrane on the length scale of tens of microns and on the time scale of minutes. And it also illustrates the challenge that we still face as biophysicists, as whole cell modelers, to try to understand these coordinated shape changes from first principles. And most of those shape changes are driven by the polymerization of actin filaments, which are schematized in this diagram of a cell. Actin filaments produce force on a membrane by polymerizing, adding subunits onto one end called the plus end, which can advance the position of, for example, the cellular membrane. And flexible actin filaments can produce more force if another protein complex called the ARP23 complex binds to the side of the pre-existing actin filament and nucleates the growth of a new second actin filament at a defined angle. And that sets up a polarized branched growing array of filaments producing directional force. And the role of these branched actin filaments is best understood in the context of cell migration, where on the length scale of a single branch, the plasma membrane is essentially flat. But equally important is the role of these branched actin filaments at membrane trafficking processes throughout the cell, both at the cell interior and on the plasma membrane. And this presents a unique geometrical challenge for the branched actin filaments because they have to deform a continuously curving membrane, and the attachments have to work out such that filaments which are polymerizing in one direction have to move the direction of the membrane in the opposite direction. And so we use clathrin-mediated endocytosis as a representative model system to study this general question of how branched actin participates in force production during membrane trafficking. So as we briefly heard yesterday, during clathrin-mediated endocytosis, the cell's plasma membrane is deformed from a flat sheet into a spherical vesicle over the course of a minute in order to internalize transmembrane receptors and extracellular material. In mammalian cells, the plasma membrane is under tension, and this serves as a source of physical resistance to those shape changes. And so a host of cellular proteins, including the clathrin coat and components of the branched actin cytoskeleton, will collectively pull and pinch on the plasma membrane until a neck forms, at which point GTPases called dynamin can wrap around the neck of the pit to carry out scission and create an endocytic vesicle, which will get delivered down into the interior of the cell. In cells with high plasma membrane tension, when actin polymerization is dysfunctional, most endocytic pits get stalled at this U-shaped intermediate, shown in these electron micrographs of cells, of clathrin-coated pits in cells. Whereas when actin polymerization is functional, most endocytic pits advance to this late stage pinched shape. And these types of experiments allow us to infer that the actin cytoskeleton is required for the shape change from this U-shaped intermediate to this late stage pinch shape. But it doesn't tell us the molecular mechanism by which these actin networks can produce force in a stochastic environment to advance the shape of this endocytic pit. So our fundamental mechanistic question is, what is the molecular mechanism by which these branched actin filaments organize in order to produce directional force in the stochastic environment of our cells against plasma membrane tension? And we think that the principles that we derive from studying actin in the context of clathrin-mediated endocytosis will apply more broadly to other membrane trafficking processes throughout the cell. And we choose endocytosis because enough quantitative measurements have been made in the context of cell biology and biochemistry in order to constrain simple computational models that will help us understand the mechanism. So for example, the location of the proteins that activate this branching ARP2/3 complex tend to reside in a ring around the base of this endocytic pit, whereas the proteins that link the actin cytoskeleton to the plasma membrane tend to reside in the curved clathrin coat. And so I reasoned that these two spatial constraints, combined with the knowledge that we have about the mechanics and kinetics of branched actin filament polymerization, would allow me to set up a simple biophysical model, and the simulations would give us predictions about the mechanism of action, which then I could test in collaboration with other postdocs in the lab who are doing high-resolution microscopy, so that we can iterate between experiments and modeling to get at more and more experimentally accessible, but also hopefully predictive models of the mechanism of action. So in our minimal model, we use the minimal actin machinery required to carry out endocytosis. So for example, we include these ARP2/3 complexes, which reside in a ring around the base of the plasma membrane, the branching factors. We include actin linking proteins, which are embedded in the curved clathrin coat. The endocytic pit itself, which is simplified to be a solid object that resists internalization elastically. And actin filaments, which can bind and unbind the endocytic pit, grow, become stochastically capped, and if they happen to encounter this active ARP2/3 complex at the base of the endocytic pit, will bind the ARP2/3 complex and nucleate the growth of a new branched actin filament at a defined angle. For this part of the model, we use CytoSim, which is an agent-based Brownian dynamic simulation software, which explicitly models individual protein complexes, actin filaments, and surfaces. And it's a stochastic model. And importantly, for a model of this size, a large number of parameters are required from kinetics, biophysics, et cetera. And luckily, and I think perhaps uniquely for a cell biological process of this size, enough quantitative information has been obtained both for endocytosis and for the actin cytoskeleton in order to constrain these models to make them predictive. So I'll give a couple of examples for the parameters that we measure. For example, to estimate the resistance to internalization, we use a continuum plasma membrane mechanics model and measure the membrane tension in our cells. And to count the numbers of molecules of some of these proteins at sites of endocytosis. In the experimental side of my work, which I won't describe today, I developed a method to use fluorescence microscopy to count the numbers of molecules of some of these key proteins at sites of mammalian endocytosis. So we run 3D stochastic simulations using CytoSim, and then we visualize the simulations in collaboration with the Allen Institute for Cell Science using an interactive 3D visualization tool that they developed online called Simularium. You can visit this if you want to take a snapshot of this QR code, or I'll show you a live demo now. And why don't I hide this video panel too? These floating meeting controls. Okay, so let's see if I can navigate over to it. Here we are. Let's make it larger. F11. Okay, so here we're looking at the inside of the plasma membrane in gray. And these purple spheres are this activated ARP2/3 complex residing in a ring at the base of the pit. And these red spheres are the actin linking proteins embedded in the curved clathrin coat. And we start with a number of linear actin filaments, which are going to seed the growth of new branched actin filaments if they hit these branching factors up here. So let's just play the simulation and see what happens. So what we find is that from an initially disorganized array of actin filaments, an emergent architecture arises in which the filaments that are doing the most work, which are growing between the clathrin coat and the base of the pit, are asymmetrically oriented. What I mean by that is that, let's get a good view over here. So the filaments that are doing the work tend to be oriented towards the base of the plasma membrane or are parallel to the plasma membrane rather than growing in other directions. I'll play this one more time. So they start out disorganized, and then over time, more and more of the actin filaments are sort of growing up over here or parallel to the plasma membrane. And this allows this network to internalize the endocytic pit against physiologically calibrated values of membrane tension, which lets us conclude that this minimal model is sufficient to understand how much force the actin cytoskeleton needs to produce to advance mammalian endocytosis. Okay, let's close that. Close that. Advance. Okay, so the mechanism of action for this self-organization we think is one of stochastic binding and selection. Namely that imagine you have two actin filaments binding and unbinding the endocytic pit in random directions. The filament that is growing away from the pit doesn't do very much, but the filament that happens to be growing towards the base of the pit encounters active ARP2/3 complex, which nucleates the growth of another branched actin filament and another branched actin filament, so that over time you select for filaments that are best oriented to producing force in a manner that allows for directional force production. So the stochastic binding and selection are a mechanism by which you eventually create this polarized branched actin network from a disorganized network. The biggest surprise that came from these simulations was that how the actin filaments were producing force. So rather than growing directly into the base of the pit, instead we saw that many of the filaments bent between their attachment sites in the coat and the base of the pit. And I thought this might be a new way for the actin filaments to be producing force. So this remained an untested prediction until Daniel Servas, another postdoc in the lab, started doing cryo-electron tomography, which is just beautifully introduced, on mammalian cells, which he grows on EM grids and then flash freezes and puts under electron microscope. And so these tomograms beautifully show individual endocytic sites, which I've outlined here, and all of the actin filaments surrounding the sites of endocytosis. So as we scan in Z through the endocytic site, you can make out all of the individual actin filaments at this particular clathrin-coated pit. And indeed, in most of the tomograms that he looks at, he sees long bent actin filaments which stretch between their attachment sites in the coat and the base of the pit, verifying that these bent actin filaments participate in endocytosis. And in order to understand their mechanism of action, I went back to the simulations and plotted for the simulations how much elastic energy is stored in these bent actin filaments over time, compared to how much energy is required to internalize these endocytic pits in the first place, which shows that there's at least enough elastic energy stored in these bent actin filaments to carry out endocytosis. Put another way, just like the pole of a pole vaulter stores elastic energy before it straightens out to produce, to do mechanical work, so too do these bent actin filaments slowly unbend in order to ratchet the endocytic pit inward against fluctuations in membrane tension. So we conclude that these bent actin filaments are storing elastic energy in order to internalize these endocytic pits. So in summary for this part of the model, we showed that a minimal actin network is sufficient to internalize the endocytic pit against physiologically calibrated values of plasma membrane tension. Based on the location of the actin nucleating proteins and the actin linking proteins, the network self-organizes into a polarized array, which mostly grows in this direction and pulls the endocytic pit in this direction. Bent actin filaments store elastic energy in a manner proportional to the amount of resistance. So as resistance increases, the filaments bend more and store more energy and produce more force. So that's a form of resilience that the network carries out. And this feedback between this model and other experimental measurements throughout the lab allowed us to address a number of mechanistic questions and other for this minimal system, and we added other actin binding proteins. So it let us make predictions at this particular length scale, but there were a number of other mechanistic questions that we had that we couldn't access with this particular model. So for example, we modeled the actin filaments as linear elastic rods, but in reality, they're helical, and the helicity of the filaments should change both their geometry and how much elastic energy they can store. And we modeled the endocytic pit as a solid object that resists internalization, when in reality, it's a continuously curving substrate that probably interacts dynamically with the changing architecture of the actin network. So both of these questions we couldn't access with our current modeling framework. So I found myself wishing that there was a way to connect this simulation framework with other existing simulation frameworks at different length scales in order to interrogate these mechanisms at different length scales. And that wasn't possible for me until I met Eran last year, who was developing a framework to stitch together different simulation platforms.
All right, we're gonna step away from actin for a little bit in order to introduce this framework called Vivarium, which I developed in my recent postdoc at Stanford. You'll notice in Marcus Covert's lab, whole cell modeling. Vivarium is a software we developed to handle the complexity of whole cell modeling, in which you need different modules, different methodologies, different modeling types, and to combine them in a flexible way that many people can contribute to. So I'm going to show you a little bit about the framework, and then Matt's going to come back up and we'll talk about the plan for modular modeling of cytoskeleton. Okay, so to start, look over here. Vivarium is conceptualized as a type of interface protocol. It's not a model in itself, but it's a framework for models to interact with one another. Kind of, I'm inspired by the internet, IPTCP, the internet protocol, which allows, in this case, us to connect many separate models, simulators, and data together in a large, complex, and open-ended network that you can continually build upon. So, for example, you might have a no. How do I get there it is. Okay, so you might have a multi-scale model of cells. They have some processes inside and down to molecular networks. If you have a hypothesis about a mechanism that's missing that might improve your simulation results, you can put it in one of these boxes, plug it in, and run the simulation. At least that was the thought behind it. So let me introduce some terminology. The basic elements of Vivarium are processes and stores. Processes are the update functions. This is where the simulators live, or the models live, that read the states and update the simulation. They're parameterized. You can pass in like a network or the constants. And then there's ports, which are where the variables are read, inputs and outputs to the model. Next, there's stores, which is where the states are held. They're external to the simulator. And they have a schema, which is kind of a type system that makes sure that when you connect your models up, it's consistent. You're connecting similar types to similar types, and it can help with the composition. So when you put these together, shown here on the right, this is what we call a composite simulator, composite model if it's been parameterized. And you can see here that the signaling module, the signaling process, and the metabolism process are linked through the proteins, which if they have overlapping proteins, we'll let them to be coupled and run together. Okay, just a very, very simple demo to break it down. This is not the complexity of whole cell modeling, but it helps us comprehend what's going on here. So imagine a process, TX is transcription. It has two ports, a DNA port and an RNA port. The DNA port is connected to a DNA store. RNA port connects to the RNA store. You can have many different RNA or DNA inside of these stores. So across the ports, you can have many different variables flowing across. In this case, just one. We have a RNA called C, a DNA called G. You can see it run over time, back over here. So when you simulate just this lone process, you can see that that the DNA is constant, the mRNA is expressed. There's also a decay rate in here. So this is a little bit more than transcription. It's also degradation. And so over a few thousand seconds, it reaches the steady state. Okay, next, we add a translation module. It reads the RNA, which lets these two get coupled. Oops, sorry, I keep on clicking the wrong button. So you can see here, same thing we saw before, the RNA increases, but now we also have proteins increase. These are ODEs in this case, and I'm going to add more complexity soon. Okay, here's how it runs forward in time. Sing Zan's discrete simulation engine actually got me very excited. We also have, it's this Vivarium's discrete event simulation engine. So you can, the process will tell you what its preferred time step is, and this can also be adaptive. So over time, the time step can change in this stochastic case. So sorry guys. The way that this flows, you get the state at the start of a time step, runs for some time, and then passes an update to update the state at the end, which then the other process might see if it reads the state at a later time. You can see here time kind of progressing. This is the front, this is the present moving to the right. Okay, there's also a second subclass of process called steps. These are non-time dependent processes. Started off almost like an afterthought, but turns out very, very critical for modeling. These run between the dynamic processes. So dynamic processes run, and in the between time steps, we run these steps. They run in a DAG or directed acyclic graph, so that these can run in parallel if they're not dependent on each other. So in this case, mass and shape will run in parallel, pass their updates, and once they're complete, we can run a division step that will determine whether it's time to divide. Okay, adding a little bit of complexity. So this is meant to be an incremental framework that you can start and add complexity as you move along. Here we've switched out the transcription process, continuous deterministic transcription process, with a stochastic one. This operates in counts. So DNA is now in counts, the RNA is in counts. Translation is still this continuous process that operates in concentrations. So here you see an example of one of these step processes that runs between, and it converts counts to concentrations, which lets these two couple. So you can see that you have this concentration RNA, which at the end of every time step reads the counts, updates the concentrations. Okay, we also support hierarchical modeling using a formal system called bigraphs. This is nodes within other nodes. So this is very common in biology, organelles within cells embedded. And so our formal framework and our systems for simulation should reflect this as well. Okay, here's the same system as before, showing how we can embed it. So here we took the same system, we put it inside of a store called 0. This is just the ID of this particular cell. That goes itself into another store. That's all the cells going here. And then you can have more simulators operating on the state up over here. So we can do multi-scale. In this case, we have a multi-body physics engine that reads all the states in here, and a diffusion process, 2D diffusion, that can read some the external fields and dimensions of the fields, and it'll update the states of what's going on in here. So you can see a cell, and then it's in an environment. So all of what I showed you before is now inside of here. Okay, the hierarchy itself can be updated over time. Again, happens all the time in biology. For example, you might have an environment with an agent inside. Division's triggered. Now you have 2. There's new processes need to be made to do that. The agent A2 might engulf A3, so A3 goes inside endocytosis, and then lysis. A3 bursts, and you end up with a similar configuration to before. This happens live during the simulation graph rewrites. Okay, so before we have a single cell division, this division process is reading mass in here. And when it's time to divide, it passes an update over here. It tells it to divide. And this is in the step after, the time step after division. You can see that we now have two cells, 1 and 2. They all have their own internal DNA, RNA proteins, and their own processes that, and this can all run in parallel. Okay, and here's the result. The same system I showed as I built up complexity. Now each one of these cells has its own hybrid stochastic deterministic system operating inside, triggers division, and you get a multi-cell simulation. Okay, after doing this, we thought, hey, let's see how far we can push it. Ideally, we don't reinvent the wheel every time. There's people who've developed sophisticated software for different kinds of simulations. So we want to reuse that as much as possible. Here are some examples of simulators that we just want to reuse, put it into Vivarium, wrap it up in one of these processes and run them in parallel. So as a demo to this, Cobra is an FBA simulator. Here we have a module. We have a few adapters that let it translate between different types of simulators. And we have the stores that correspond to Cobra. So this is like a composite module that we can take in and allows it to be compatible with other simulators. There's also pip installable for just, you can pull it into your notebook and run it as is, put in different models to configure it. Next, we have Bioscrape. This is a chemical reaction network simulator. You can run deterministic or stochastic simulations. And finally, we have this composite for the environment. Multibody is a physics engine. It's actually a video game physics engine for apps, which I put into here and try to reuse it. I don't want to create my own physics engine. And diffusion is for the two-dimensional diffusion on a lattice. Put it all together, we have a more complex model than what I showed you before. So we have the Cobra. This is flux balance analysis, a chemical reaction network, added a division process that reads the mass through the right over in here. And here's the environment, the top right, and some additional converters that let us read the external state of the field so that the chemical reaction network and flux balance analysis knows the external state. Some results of this. This has elements of all the simulators combined. So this is a massive hybrid simulation. We also were added to the biosimulators pipeline. See over here. This is the database for simulators that Jonathan Carr introduced yesterday. And there's about 60 different simulators available here. They share an API, which means that all of these can now be plugged into Vivarium and combined into hybrid simulations. Okay, just a little bit more. We published a paper on E. coli in the Covert Lab. Here it is in Science 2 years ago. And big effort, hard to modify, not very flexible. And thought, let's reconstitute this entirely in Vivarium to allow for more flexibility and modularity. And so with the help of some students in the bioengineering department at Stanford, we remade E. coli entirely in Vivarium. You can see all the processes down here. Each of these is a different type of simulation. We have metabolism is flux balance analysis. Complexation is stochastic simulation. We have chromosomes are an agent-based simulation. And all of our different states up here. You can see the DNA, replisomes, chromosome, etc. Here we can see the distribution of masses. And then we can also have some fun and plug it into the multiscale framework, the multi-cell framework that I showed you before. So reusing the same environment as before. Each one of these is an E. coli whole cell model. And we ran this on Google Cloud. took quite a lot of compute. And let's see, AMP-C. So here we can tag proteins as well. So here we're tagging AMP-C, and you can see just due to the stochasticity of the simulation that there's a results in the heterogeneous population. Again. Okay, so growing on. There we go, growing on glucose. So you can see glucose being depleted first slowly and then a little bit faster towards the end of the simulation. There it is, same simulation again, but tagging AMP-C. And here it grows. We can do this for the many thousands of proteins or genes or growth rate. The beauty of simulation is we can tag whatever we want. So tagging growth rate was a fun one. Okay, back to our collaboration.
So after I saw Eran talk about this modular framework, I thought this would be a fantastic way for us to do multi-scale actin simulations for endocytosis so that we don't have to reinvent each length scale of simulator. So our long-term goal is to take the endocytic actin simulation that I described, which is in CytoSim, and link it up to other simulators at different length scales for both the actin cytoskeleton and other components. Eventually, we'd like to zoom in and out of the simulation, and as you zoom in, you switch to a different simulation framework and asking a different mechanistic question. And so our collaboration started out as a secret side project between Eran and members of the Allen Institute for Cell Science, whom I had met through using our endocytic model as an initial use case for their Simularium interactive viewer. So Eran and these members of the Allen Institute decided to use actin filaments as their first use case to see if within the Allen Institute's online web-based framework, if they could start to house different simulations of the actin filaments and then tie them together using Vivarium. So once they succeeded in tying two actin simulators together, they came to me and said, we have this cool tool. Do you have any biological questions that might be addressable by multi-scale modeling? And I said, yes, I do. And so that's when I joined the project. So the simplest question that we could think of to start with is, what are the other mechanistic consequences of these bent actin filaments that I just described? So for example, you have this bent filament here. It's attached at one end and it may or may not be attached at the other end. And it's bending, it's probably also twisting. We want to know what happens to its geometry and to its elastic energy storage. So as an initial simple first use case, we took the same model of a single actin filament bending. We put it into Vivarium and we ran two parallel simulations in two different simulators. So for example, if you run an actin bending simulation in CytoSim, the filament bends like an iron rod, like you might expect. And for comparison, we stepped down a length scale and we took advantage of an actin filament model that Joe Schoenberg, who spoke yesterday, created with Blair Lyons from the Allen Institute, using his particle-based Brownian dynamics simulator, READY. And so fantastically, they now have made an actin filament at subunit resolution. So each of these subunits is a different particle, and the potentials are set up so that the actin filament has a realistic helical 3D shape. So we ran our model, our simple bending model, using READY, and we can observe how the conformation of the actin filament changes in a way that's different from when it's a simple elastic rod. So here we're visualizing the actin filament in Simularium, and you see that it doesn't behave like this linear rod. So there are other modes of bending, there's twisting that's coupled with the bending in a manner that depends on how you attach the actin filaments at the end, how much you constrain their rotation or their translation. So this both let us calibrate various parameters like the stiffnesses of the two actin filaments to make sure that we had comparable actin filaments once we wanted to switch between the two of them. And it gave us some insights into how much extra elastic energy the bent actin filaments store when they twist. So it turns out they store about twice as much elastic energy And they rotate the whole network along with it because the filament is twisting. So this is the kind of feature that we want to add into our endocytosis model. And so the next step in this initial stage of our collaboration is simply to stitch together these two simple models of the actin filament, just to have a proof of principle that we can switch between the two actin simulators, which Eran will describe.
Okay, so here we have, you are all now familiar with the Vivarium graphical language. We have our processes in rectangles and the states or in stores shown here as a database. So we have two different types of states: fibers, which in this case we have a fiber scale simulator called Median, which is comparable with cytosm. We've actually interchangeable with cytosm, in this case Median. And we have states here for the subunits, which are connected to Ready. So Ready can read the positional information about subunits and run it. And we need to be able to translate between these two. So you're all now familiar with the step processes that run between the time steps. And these allow for translation so that the same state can be read both in the subunits and the fibers. So we have a fiber to subunit adapter, which takes the fiber positions in 3D space and puts monomers in its place. So reads from fibers and then translates it to the subunits, which Ready can read. And we have the inverse, the subunit to fiber, which takes the positions of the subunits and puts back a line in 3D space so that the fibers can read it and Median or cytosm would be able to simulate it. Okay, very early stage movie here shown in Simularium of that happening. Okay, so starting off in Median, you'll see it's transition to cytosm. Here we can turn around, back to Median, back to cytosm, back to Ready. Sorry, I might have said cytosm. It was Median, Ready, Median, Ready. Okay. So there's a few things we're currently working on. This is, again, a work in progress. Ready runs at much smaller time scales. It's more computationally expensive than Median. So we don't want to translate this whole thing and run it in Ready. Rather, we want to be able to detect where it's necessary to go down to Ready, and maybe in these cases where things are highly bent and there's divergence in the prediction, which is why we did all these comparative studies of comparing the fiber scale and the subunit scale to know which pieces of this simulation need to be kind of cut out. So we have a bounding box and cut that we identify that, cut it out, convert it through our adapter, run a little simulation until things are resolved, convert it back through this adapter, and then stitch it back in so that we can resume with the fiber scale simulation. It's even more complex than that, though, because the fibers over here can exert forces into the box, and we need to be able to translate that reliably so that if there's a force coming from the left of the box, that'll be applied within the Ready simulation. So that's also something we're working on. Additionally, we want to make sure that all of the different states, including the proteins that are represented in one simulator, have a corresponding state that can be incorporated in the other simulator. So there's a process of matching like to like so that we can reliably do this process of cutting it out, including everything that would be inside the box, converting it, running it, converting back, stitching it back in. Okay, And for the future, hopefully the relatively near future, we're waiting to hear back on a grant about this. Working with Padmini Rangamani, she has this amazing deformable membrane simulator called MEM3DG. And we want to couple all of these together so that, well, Matt had mentioned that the fiber scale simulations are typically run in rigid bodies, geometries, where the membrane doesn't move, but that's not really the case. So in order to get the fibers to exert forces on the membrane, have the membrane deform, that changes the geometry for the fibers. We also have, you can see here, we have these different simulators at the same scale. So Cytosim and Median are comparable simulators at the same scale. Reddy and Smolden are about comparable simulators. And so we want to be able to have similar simulators so that we can compare them and contrast them and evaluate which is better in what cases, and maybe have an effort to standardize these so that through their shared interface, they can be compared and improved over time. Okay, and that's where we're headed. Thank you very much. And thank you to my funders. And also, I want to make a note that the Agmon Lab is hiring. We just started this month, beginning of the month. And I'm looking for postdocs, graduate students, software engineers interested in whole cell modeling. We're going to take Vivarium, develop it further, and integrate all the simulators in biology and make whole cell models. So please contact me if you're interested. Thank you.
Thanks. That was a really great talk. I had some questions about, I really love this illustration that you had about the switching between the different scales with the fiber at the end. And I was wondering if, I'm trying to get a sense of, are there general principles about multi-scale modeling that you can extract from that or apply? Because in some ways it seems like, it's sort of general, right? You're going from some sort of continuum model to a discrete model, or is it like, is there any way to automate that? Or do you really have to, like, for each specific case, like build in kind of the box and stuff, how you're talking about, you need to assess like where it's going to fail. So yeah, I'd be really curious about your opinion on how generalizable could you make that to any aspect of cell biology?
So, there's some things that can help. For example, making sure to specify the types at that the scale that the given simulator can work with. So like, let's say the fiber scale, there's a type, it's a lines in 3D space, right? And the subunit is these positions of particles in 3D space and their linkages. So if we can specify that type, we can know what needs to be connected to translate between them. Mm-hmm. So if we have a different simulator also specifies the same types, we know that they can be exchanged easily. And we can create these adapters that translate from one type to another. Those can be reused if we have simulators with the same types. That being said, I think we need a lot of adapters to go for all of biology. But if we have a shared framework, we can fiber to subunit is something that we've done now. So put that online. It's pip installable. Should be able to reuse that. And I think over time we will find that we have a finite number of adapters to make that possible. There's other principles too, the bounding box and cutting. That's something I'm not sure is entirely generalizable, but same thing is, let's just look at all the cases, figure out what needs to happen, make sure that these are reusable and shareable, and reuse them as much as possible. And I think that we will see over time that we converge on a set of adapters that are needed.
Okay, that I'll agree. That was awesome. It's so cool. I guess my first question goes kind of to the first part of the talk where we were looking at. So I was just sort of wondering about whether you had looked at the way that things like the number of those ARP2/3 things in the ring, it sort of struck me that if I have cells where the membrane's under different tension, I might have different like results or different types of dynamics. If, for instance, it's under less tension, but you have the same number of ARP2/3s, you might take boom, it might just be like ripping the thing. And then if it's like under way more tension, and that might be something that happens, I guess, in real cells, like every mammalian cell in our bodies or every human cell might not have the same membrane tension depending on the local environment. So I was just curious if you looked at that and if there's any like work that's been done to sort of see, is there a difference in those parameters between cells with different sort of tension properties?
Yeah, so the membrane tension and ARP2/3 complex activity are two of the experimental knobs that we can turn in a somewhat quantitative manner. So there's a specific small molecule that you can titrate down the activity of the ARP2/3 complex. You can change membrane tension by inflating the cells by changing the osmolarity of the medium. And so for the simulations, we have this great phase diagram of, as a function of ARP2/3 complex activity and membrane tension, how much force do you produce? How far do you get? And it matches really nicely with the membrane tension variation experiments that my lab mate Charlotte Kaplan did in tandem with tuning down ARP2/3 complex activity, which is something that I did on the experimental side of my project. And so just as you might expect, when membrane tension is lower, you need like a small amount of ARP2/3 complex activity is sufficient. When membrane tension is higher, you need all of those ARP2/3 complexes. And for comparison, the force regime for mammalian cells is about 200 times lower than the amount of force you need to do yeast endocytosis, which Julian in the lab or in the audience studies. And so there you have a bit more ARP2/3 complex, and you have different ways to organize the actin network.
Very cool.
I guess I'll ask, this is sort of nuts and bolts questions, but I was wondering about the sort of the time stepping stuff that you were talking about. And I was curious how automated can that be in terms of the, the decisions about the time steps and the feedback between the different levels? Does it work better if you can assume certain time scale separation, or does that not matter?
So time scale separation can help because when things are coupled and they influence each other, you can get like what's called like stiff situations. And well, let me actually step back a bit. We found that having a central coordinator of the time steps is probably not something we can do because there isn't enough information about the individual simulators, about their sensitivities and what they respond to. So we made a design decision that the individual process itself has to provide its time stepping information.
Mm-hmm.
So if you have a Gillespie simulator, you see the state at the start of the time step before it runs. It tells you how long it needs to run for, and then the engine says, okay, you can have that, and it runs for that long. And you can do all kinds of adaptive time stepping in that way. But when you're combining Boolean systems with ODEs,
Mm-hmm.
you're not going to have a central timekeeping mechanism because these are just very different. But a lot of simulators actually have that code that determine how long they need. And so we just have to expose that part and ask the simulator the question. Regarding timescale separation, I think we have to be careful. There's some domain expertise that needs to go in. This thing, it's not quite automatic.
Mm-hmm.
So you have to know your system.
Mm-hmm. Well, you're building it, so that's cool.
So I'll ask a follow-up question about the Vivarium thing. This is the first, maybe ask two. First one's really quick. So it seems like a lot of like what was flowing between some of these things would be things like volume, and that's all. I guess that's really easy to put in there. Like you basically like as the thing grows, it's going to get larger in volume, and then that's going to change the parameters of the stochastic simulation. For instance, it's going to make collision less likely or something like that.
Mhm.
And so do you, when you're building one of these things, I guess if you're building one of them, you have to kind of know about all of those like ways that you need to hook up and think about how everything needs to kind of be composited. Because I might not immediately think, oh, the stochastic simulator needs to know volume, but it does. So anyway, I was just throwing that out there. Like, is
Yeah.
there like, did you provide help with that, or is that something that anyway?
So yeah, we want to help people, right? Composition is hard, and so any kind of assistance that we can provide, I'd want to do that. So if you have a following your example, if you have a stochastic simulator, there's going to be a port for volume,
I see.
and that has to be plugged in. And we're not going to let you run a simulation unless you plug that in. So that's a little bit of assistance, like, hey, where's the volume? And so if you know that where the volume is stored, you can plug that in. It also, there's typing. So the volume is specifically in a certain type. You might have milliliters or and so if you have a state that's stored femtoliters, I need to make sure that the types match. That's a little bit more of assistance that we can help you with, like, this has to be compatible. You can have an adapter that lets you convert between them. So that's a little bit of help that you can do. But the person who provides the process, the module, has to be very clear about the inputs and types.
Yeah, so the other question I had is that this is maybe a little bit too technical, but it's sort of related to what I asked Jonathan about yesterday. So some of your the sort of simulation engines you're talking about have these agent-based simulators that deal with like extremely large combinatorially complex state spaces where you might have 10 to the 23rd, 10 to the 26th possible states. So you wouldn't be able to store those in containers where every single one of those species has a concentration or number. So does that mean that in those cases, those plug up to storage containers that are agent-based storage containers that then plug? Or I'm just curious how you solve that problem if you use these quote unquote network-free simulators.
So this is in a way kind of so you could have a network-free. Let's say we take BioNetGen and we put in one of these processes. I showed a slide in which the network itself is configurable during runtime.
Yeah.
So let's say we plug in BioNetGen. This is something I'd love to try, and my office is right next to Michael Blinoff, so we'll possibly talk about it soon. Michael, if you're out there, we should talk about it. But so if BioNetGen says, hey, we have a new state that arose, that should trigger a new store to be added. And then we can run forward that way. So that's one possible solution. Don't pre-specify all the states, but rather use the graph rewrite rules to expand as needed.
We'll talk, because there's actually results on the fact that that approach can be pretty inefficient compared to the agent-based approach that the simulators themselves use. That's why they do it that way. So we can talk about that later.
Okay, yeah, let's.
You guys still have, should we go?
I'm good. We can open it up.
Ready? Okay, so I have a couple questions and then we have some in the audience. So I'm going to represent the stupid people who are thinking about something that they're afraid to ask. Or maybe I'm just the only stupid person. Not sure. Starting with Matt, when you were showing us the filament as basically just a rod or a line bent, elastic energy. Intuitively, the way you have pictured it to be, it seems like it's pushing the wrong way. It's not extracting, it's pushing. So then we get to the subunits. That's a whole different story. And I think, okay, that's the solution. But then in the end, I got confused. Darren, what you did is you took the filament, you made it into subunits, and then in the end, you switched it back to put in the little square. Now we're going to put it back as a filament after having cycled it through a more complex subunit. So I thought that defeated the purpose. Now I'm just really confused.
Okay.
Okay.
So the first question, we wondered the same thing. Are these bent actin filaments going to push the pit back up or extend it? So I just ran an in silico experiment as an experimentalist and said, let's let the actin filaments bend and then release membrane tension to cut off the spring, stop actin filaments from growing, and just look in the simulation. Does the pit go up or does it go down? And the filaments straighten out and the pit goes down. So observationally, we see that the straightening filaments are leading to increased internalization. We think that's probably because of an asymmetry in the attachments and the attachment geometry of at the coat versus on the plasma membrane. And then for your second question about what changes on the filament level once we've gone from subunit to filament back, we think that there will probably be changes in the geometry. So part of the let's say distal part of the actin filaments may have rotated, et cetera. And so you want the rest of the network to update.
Right, so you're getting a better idea, representation of the spatial arrangement.
Yeah. And the other thing that could happen when you twist the filaments too much is that they break apart. And so this is part of the disassembly mechanism that can happen just by mechanics. So if they break apart, then we now have two separate agents, et cetera.
David, I don't know if you're talking at the moment, but we can't hear what's going on in the auditorium.
But now that we have, we want to make that space compatible with Vivarium and hook it up to some other simulators where we have. What would you recommend as a process again? You have tutorials, should I come to your lab? Like, how would you do that for someone who hasn't done it?
Yeah, I've worked with a lot of different groups at this point to help get their simulator in. Up till now, it's been mostly sit down with me, and do we have to talk about the API of your simulator? What are the relevant states that have to be exposed? What we do is a process is like a wrapper around the simulator, so it can invoke your simulator. It reads the states, invokes an instance of a simulation, runs it for some time, needs to read out the results. So far, I have not found a simulator that I can't put in one of these wrappers, but it's some software engineering and sometimes not very efficient. I do have a lot of tutorials about it, Jupyter notebooks online. If you find my website, they're linked there. You can email me and link there. But yeah, and probably at this point, you'll need to bug me, so please bug me. And I really enjoy it. And we can sit down and talk with the developers about, really it's hard for me actually to build a wrapper around a simulator without talking to the developer, because there's really nuanced differences in the API that make it hard. So just talking about like, hey, how do I access the state? Or right now there's at present there's it's a Python API, but we're working on making it language agnostic, have like a general interface protocol. But for the current version of Vivarium, you need a Python API that we could call. But we do have examples that within Python we can call something in a different simulator. It's a little bit harder and harder to interpret and harder to work with, but it's possible to build a wrapper that just invokes an executable. Or so.
So I had a question too. So great talk. My question was, it's probably more biological. So if you look at listeria being propelled by actin bundles, then the listeria rotates. I was wondering whether there's any rotational sort of motions which sort of twists off the endocytosis sort of vesicle out here, whether there's any experimental evidence for it. But my second question is, could the simulation have predicted it without you putting it in by hand?
Yeah, so a proxy to rotation is like an asymmetry in forces from ARP23 complex. So that's something that we see in the simulation, that often there's more actin ARP23 complex on one side than the other. And then another postdoc in the Drubin lab, Manjin, looked through super resolution microscopy and indeed saw that often the branched actin was sort of pocketed more on one side than the other. And so they're hypothesizing that this could cause rotational twists. For this particular cytosine simulation, I turned off rotational motion just for simplicity. And so we missed that prediction. But once we have a realistic 3D membrane, I bet that we will see rotation. That can help with decision process, et cetera.
I have a question about the regulation of like mechanical strain in the filament network. And so I was wondering, like naively, I see a buckling filament like actin filament. So I think of severing. So I was wondering if you see like a role for like cofilin, kind of like coming in, severing filaments that are too bent.
Yeah, exactly. That's I think the exciting next step that we want to do in collaboration with the cofilin people, which we've started. So just in the minimal model, we plotted based on how much they're bending, what fraction of the filaments would be severing on their own if cytosine let you do that. And it's like 1 or 2% of the filaments are so bent that they should just break apart. As you might know, bent filaments favor cofilin binding. So there's a positive feedback loop. Once they start bending, you get more cofilin, that's going to sever the filaments faster. So that'll be a really fun thing to add that we haven't done yet. But that's part of our general hypothesis about how the shape, the compression, the extent of compression of the actin filaments can be a signal about, are you at the beginning of endocytosis or are you at the end? And so maybe there's a disassembly mechanism that's sort of self-perpetuating once we start adding cofilin. The reason why we didn't add cofilin in this framework is that these actin filaments don't have an ATP state. And from my perspective was that the preference of cofilin binding just the old filaments, the ADP filaments, was the critical thing about them. And without that assumption, I thought it wouldn't be predictive. So once we add that, then we can start adding cofilin.
Great, thank you.
Guys, that was great. And you guys will surely answer them. Can you put up your QR code again? I didn't get it. So it's lunchtime.
Okay, we'll see you back here at 1:30, okay?
Well, I think that was short and sharp, a nice ending to everything. I think John and Robert as the loyal stickers here. Oh, and Michael too. Lunch, guys, you know the form. Well, sorry, Robert, it's supper time, dinner time, I'd imagine. Perhaps even a glass of wine time. It is Friday at this side of the Atlantic, after all. And
1 o'clock.
Wine o'clock. I'm certainly going to be having a small glass a little bit later. I'll try and do it discreetly so I don't look like.
Let's all come back staggering drunk.
Challenge accepted, Wallace. Challenge accepted. Should we reconvene at past? That would be lovely. We've got the final sort of final little bits for you. I think it's all of you are likely to be interested in Matthew Buchner, who's the from the NSF, and then we've got the 3 concurrent sessions. But clearly, it's likely to be Matthew who's going to be of interest at 1:30. See you then. If you want to leave Zoom, that's fine. I'll sort of hang around, but I may zip away just to get myself a glass of wine and something to eat.